All right, so thanks for coming. This is the first seminar of the spring 2017 COBRA series. So this seminar is for the students to kind of get introduced to the research that's going to be happening this summer. So we have Dr. Richardson, Baldus, Budenr, and Parker, it was a random order, talking about what they're going to be doing. So each of them will be able to take two coastal students this summer for research. And Richardson's going to kind of start us off by giving you some information on how to apply, what's going to happen with the program, and just kind of what to expect. So Dr. Richardson, get us started.
All right. So I want to talk about the INBRE program and how it relates to you all. And first, just give you some information. First off, we have a website. And if you're trying to find it, ccuinbre.wordpress.com, or just Google CCU INBRE, and it will pop up. Okay? And everything I'm going to talk about, it's there someplace in one of the pages. We also have a Twitter site, so you can follow us with Twitter, because Brian is really good about reminding us about staying on social media. So we have all this stuff. As a matter of fact, I think I set up the Twitter, but Brian is in charge of the COBRA series, and he set up the YouTube page, so you can actually relive this whole experience later on tonight, or when we post it. So, I mean, you can have fun.
Yeah, slow down, later on tonight.
Well, maybe later on next week. All right, so let's talk about some of the goals for the SCoRE program. The SCoRE stands for Summer Coastal Research Experience. I mean, you can score with coastal, and that's our motto for this beautiful program. So number one, we're going to build on establishing multidisciplinary research networks with a scientific focus to strengthen the lead and partner institutions in biomedical research expertise and infrastructure. Paraphrase, we're going to do some biomedical research this summer, and we're going to work with other institutions and use some of their facilities. Build and increase research base and capacity by providing support to faculty. They're going to give us money so we can pay for stipends and also pay for supplies. Provide research opportunities for students from primary and undergraduate institutions. Normally, you've got to go to a very large university if you want to be able to get a lot of grant money to fund student research. So we're going to give you some money so you can actually hire some students and have them come in and do some research. And then enhance science and technology knowledge of the state's workforce. This is actually one of the state goals that we tagged in here so everyone is happy. So let's talk about the application process. So you can actually download the application now. It's at this website right here. And this right over here is what that website looks like. And you just click on this right here, right underneath Uncle Sam, and it will download the doc file for you to go ahead and fill in. So these files need to be due by March 10th, all right? And you email them to me, and if you look at the first page, it'll have my email on it. And what I do is I take the files, I write off some stuff in Excel sheet to keep track of that, and then I will forward it to the individual faculty member you're interested in working with, all right? And then you will then interview with the faculty sometime after March 10th, and that will be set up between you and the faculty. And then the faculty member will make a decision near the end of March about whom they would like to take. Now, let's talk about qualifications. First off, you must be 16 years old or older. This is based on university policy and also state law and working in the lab. Believe it or not, we've already had a 12-year-old express interest in trying to do this summer. And they were very upset when I told them they couldn't. We actually do take high school students as well, and we'll talk about that. Each faculty member will have their own requirements as well, what they're looking for. So it's important that once you've figured out who you want to do research with, go talk to them. Find out what they're looking for. All right? So speak to them. So here are our 4 faculty members, and this is just a little slide kind of showing you what we're going to do. So each faculty member is going to take 1 CCU undergraduate. They're also going to take 1 high school student. After that, they can choose if they want to take another CCU student or undergrad or a high school student. So it's going to be up to that individual faculty member to decide what they want to do. Now, for the undergraduates, the program will start June the 7th. and it will end August 11th. Your pay is about $4,000 for the 10-week program, basically $400 a week. And that first week, you're going to be trained on mentoring skills and some other things to get you ready for when we really kind of get going in the second week with our high school students and everyone else. Over here, this is the page for the SCoRE program, in case you're interested, and the website is right there. I'm going to skip this part, but this is for our high school students. Do we have anyone from high school here? Okay, one. So no, I will not skip this part. What high school do you go to? Scholars?
Scholars.
Okay. So yours starts June the 14th, and you're only going for 9 weeks. Your pay is $3,600. You're getting the same $400, but you don't do the first week. And that's to give our undergraduates a head start of getting ready for what's going on there. And pretty much the same requirements as I mentioned before for you. But you also we'll be having a high school interview day where you also come with your parents. And that way your parents can talk to the faculty and ask questions, and you will also have your interview at that time. So where is this research going to be done? It's going to be done under the rainbow at the beautiful Swain building, except if you're Budenr. Budenr wants to be difficult. And where are you doing yours? At the new science building?
Yeah. Okay.
Okay. So if you're with Budenr, you're actually here. I didn't know that until earlier today. That would have been nice. My slide's all wrong. I'm hurt.
Can I ask this question?
So what else are you going to get? Hands-on mentoring for research scientists. So here, you're going to work with the research faculty full time. You're also going to have hopefully someone there who's pretty experienced in the lab. It's a pretty interesting experience. So I kind of threw some pictures up of, look at these happy students working with faculty. Dr. Parker, you didn't even know I had this, did you?
Mysterious.
Yeah. And I mean, look at the happiness of the students. Is Rebecca here? No.
You can't tell. That's a snake.
Yeah, she's
That's a
kissing
snake,
a snake.
that she's kissing. just put it. Yeah.
All right. So let's talk about some of the other things that the program will provide, and I want to talk to you about COBRA. All right. So COBRA is one of the programs, and that is run by Dr. Wakefield. And it's the Coastal Biomedical Research Activity Series that we do year-round, where we have guest lecturers come in, and they'll talk to you, and we post those lectures on the YouTube site. We also have, we have them go to MUSC. We've had MDs, different professionals come in and talk. And we also kind of tie it in with the pre-health committee work and that kind of stuff. We also do poster sessions, and we'll talk more about that because you'll be going to conferences and presenting your work. We also do a poster session at the end of the summer for our research, and then, I don't know when this year, but they'll tell us 2 weeks before we're supposed to go, we'll do some conference in Columbia for the entire INBRE network and all of the other 10 universities that take part in this program. By the way, in case you didn't know, this is COBRA Commander. AKA Dr. Wakefield. Here's some examples of some of the professional development we do for those of you who get selected. We have mentoring. We have lab safety. We have to all do this. Then we do research ethics, our trip to MUSC, applying to professional school, resume and CV writing, cover letters and personal statements, literature searches, oral presentations, how to make a poster, poster prep, meet a doc, and then we have, of course, at the end of the term, our poster session. Well, we had, I think, about 40 to 50 people show up last summer for our poster session. A lot of family members and a few faculty and community members. All right, here's some photos I took at that session. We had a lot of people in the entranceway. We do this right in the hallways of the Swain building, and we did this on August the 12th, basically next to last day of the program. I think it was the Thursday before we finished. We also take students to conferences to present that. South Carolina Academy of Science, that's going to be a big one coming up because it's actually here on campus. So I'd encourage all faculty to see if you can get your students to get into that. We also go to the South Carolina INBRE during the summer. I've taken students to the American Society for Microbiology. I think Dr. Budenr and Dr. Wakefield went to CERMAX. So there's a lot of different places we take you guys, so you actually get some experience going to scientific conferences and presenting. As I mentioned before, the COBRA series, here it is. Our next famous speaker is Dr. Van Hoek. There he is. And I guess you're on the 17th. And then we have some other speakers coming in. We also have students. You guys come out and talk. And we usually do this before a conference, so you get the feel of what it's like to be in front of a group and take questions before you kind of go out and present at these conferences. And then finally I want to talk to you about the 4 faculty members who were selected. We have Dr. Baldus, Dr. Budenr, Dr. Parker, and then myself. I'd like to tell you that you are free to apply to each and every one of us. But you have to turn in an application for each scientist. Is that clear? So if you want to do all 4, you have to turn in 4 applications. And you probably want to make sure each one is tailored to each of the individuals you're applying to. So you want to meet with them to find out what are the skills they're looking for, that kind of stuff. And then once again, if you go to this page, you can click on these individuals and you can go to their websites and get a lot more information about what they're doing. And with that said, I'd like to introduce our next speaker, Dr. Baldus.
Okay, so there are a lot of my students in the audience. A lot of you know me already. The ones that do not know me at this point, my name is Karoline Baldus. I joined the Coastal Carolina family about 3.5 years ago. I moved down here from Nebraska, from the Midwest. Now what you're hearing is not the typical Nebraskan accent. It's actually an accent from Germany. So I've lived in the US total about 14, 15 years. So I got my Ph.D. in Nebraska, a Ph.D. in medical sciences. And after I got that Ph.D., I joined the laboratory of Dr. Wagner at the University of Nebraska Medical Center. And his laboratory is doing all kinds of cancer research, breast cancer research. That is the team that I joined. And we were looking into the role of a protein, TSG101, and its role in cancer initiation and cancer progression. And the research I want to be doing this summer is basically in continuation of the work that I've started in Nebraska, and will be done in collaboration with the Wagner Laboratory as well. So today I just want to briefly talk about TSG101. What do we know? And I really will keep it very brief because we do not have that much time. I want to tell you a little bit about a novel mouse model that we established, or that got established in the Wagner Laboratory, and want to tell you what we will be doing with it in the summer. So let's look at TSG101. TSG101 is the protein product of the tumor susceptibility gene 101. And it got first discovered during a random screen for tumor suppressors. And you can see here we have the question mark in the back. Now we'll get back to this in a little bit. But just so we are on the same page, you might know this already. I hope a lot of you know this already. A tumor suppressor gene usually codes for a protein that either inhibits cell division or negatively controls it. Right? Meaning that if we delete this gene, if we take that protein away, what we get is actually uncontrolled cell growth. And if you know anything about cancer, you know that cancer is a disease of uncontrolled cell growth. Right? So let's keep this in mind. The Wagner Laboratory at that time, long time ago, said, okay, great, there's a new tumor suppressor gene that got discovered. Let's make a mouse model. And let's delete TSG101 in that mouse model from the cells in the epithelial cells, some of the epithelial cells in the mammary gland, and then we should get a tumor. Right? And that's what they did. And they found something very interesting. They found that TSG101 is essential for cell viability. They found that if you delete TSG101 in the developing embryo in the early stages, you actually get embryonic death. So the embryo will not develop. It's essential for embryonic development, essential for the survival of adult tissue. So adult cells need TSG101 to survive. And what will happen is if you delete it out, all the cells will undergo cell cycle arrest at the G1S border and will die eventually. Now let's go back to our definition of a tumor suppressor gene, that I just told you about. And TSG101, a tumor suppressor gene? No, probably not. Because Again, tumor suppressor gene, we deleted out all the cells. We got uncontrolled cell growth and not cell death. Okay? So that work was supported later on by, again, the Wagner lab showing that TSG101 actually has weak oncogenic properties. So it can induce cancer. It's associated with cancer. They showed that it is actually overexpressed in a subset of invasive human breast cancers. And then later on, this was supported by, you can see here, lots of other researchers that actually found that TSG101 expression is actually upregulated in a variety of human cancers. And you can see, besides breast cancer, we have ovarian cancer, thyroid cancer, lung cancer, colon cancer. So definitely there is an association with TSG expression, high expression levels of TSG101, and cancer, right? So let's talk briefly about the functions of TSG101. Actually, it does have a couple different functions in the cell. I do not have time to talk about all of this in detail, but there's one thing I want to tell you in regard to the function of TSG101. It is a member of the endosomal sorting complex required for transport, the SCoRE machinery. And this SCoRE machinery is critical for the regulation of growth factor receptor expression. And some of you might recognize those growth factors, the EGF receptor, the IGF-1 receptor, the ERB2 receptor. Those are all receptor tyrosine kinases. And if you remember from some of your lectures, hopefully, then, let me bring a pointer.
You can use I left you the clicker.
This one?
Yeah, hit the big button.
The middle one?
Mm-hmm.
Okay, perfect. Thank you. So our growth factor receptors are membrane bound. They're on the surface of the cell, right? And if we have a growth factor that binds to them, if you remember them, you get a signaling cascade that triggers, in our case, because we're talking growth factor, that will trigger cell growth and will tell the cell to divide, right? Now we are getting more cells. One cell divides, we're getting 2 cells, right? Now, again, more cells, if this process here is uncontrolled, the cell division process, if we're getting too much stimulus from a growth factor receptor or the growth factors themselves, if there are too many growth factors, again, this is unregulated. This can result in a cell growth, again, the cancer topic. Okay? And in fact, blocking growth factor signaling has been one very common and valid strategy in cancer therapy. Okay? Now, knowing this association, what I've talked about before, that TSG101 functions as a member of the SCoRE machinery, that the SCoRE machinery is important for the regulation of growth factor receptor expression on the surface of the cell, we were interested in knowing what actually happens if we delete, what happens to growth factor expression, receptor expression, if we delete TSG101 out of the cell. We already know the cells will die eventually, but while the cells are still alive, in the first couple of days, what will happen? And what you see here is a Western blot. That is actually a paper that I co-authored. We published this in 2012. And what you can see here, who's familiar with Western blot? The students. Who's familiar with Western blot analysis? The results? Okay, so briefly, you see those bands here, and those bands represent the proteins that you see listed on the right-hand side. Okay? And normally for Western blot analysis, we say that the stronger the band is, the more protein we have expressed. Okay, so darker bands, more protein. And what you can see here, that first lane represents the cells that still have TSG101. And then over here, you see we have a homozygous deletion of TSG101, meaning that on no allele we actually have TSG101 left. There's no TSG101. And you can see that if we do the Western blot, if we test for protein expression in the cell, in the cells that should have TSG101, you see the band here. After deletion, it's more or less gone. And now if you look at the expression of the EGF receptor and the ERB2 receptor, 2 of those receptor tyrosine kinases, 2 of those growth factor receptors, then you can see they actually go down as well. And we try to do this in a time-dependent manner. If you delete TSG101 out of the cells, you can see that the TSG101 levels go down. And look at the EGF receptor levels, they go down as well in parallel. This was a pretty cool finding because we have here not just downregulation of one receptor, we have downregulation of 2 receptors at the same time, and 2 receptors that have been shown to be important in cancer initiation and progression of breast cancer. Okay, so really cool finding. Now, whenever you have a finding like this, in cell lines, this was done in cell lines, cell culture, you want to see is there any relevance in the living organism. That brings me to the next step. So during the last year that I was in the laboratory, they started working on a novel transgenic mouse model. And I want to talk about this briefly because there are two very cool aspects to this mouse model. First of all, the expression of TSG101 in this mouse model is tissue specific, meaning we can localize it to the mammary gland. And we do this by using a specific promoter, the MMTV promoter. So this is expressed in all epithelial cells in the mammary gland. And the second part is, and that is the cooler part, the temporal expression, meaning we can decide at any point in time, do we want to have expression of TSG, yes or no? And this part here is super important when you talk cancer research. Because think of this. If you have a protein that you think could be a target in cancer therapy, what you want to do is you want to make an organism, a model, for example, a mouse model, where you can wait for a tumor to grow. And once you have that tumor, you want to be able to turn the expression of your protein off. Because at this point, you want to observe your tumor and see, does it shrink? Does it keep on growing? What does it do? Right? And only then, if you see that, you take your protein out of the equation, out of that tumor tissue, and your tumor shrinks, that can give you a pretty good idea in regard to, can you use your protein of interest as a target in cancer therapy? Yes or no? If it shrinks, then yes. You should probably look into that. Okay, so you want to be able, in a model, in a mouse model like this, you want to be able to induce or stop expression of TSG101 at will. And we do this with a system, it's called the tetracycline inducible expression system. And I want to explain this briefly, how this works. It's pretty cool. And this is something that whoever does research with me in the summer will have to deal with this and play around with this. So let's start with the middle part here. That is the important part. We have, this is our gene of interest. So we have two, let's take a step back. For the tetracycline inducible system, we have two systems. They're two separate systems. You can do one or the other. We have the tetracycline off system and the tetracycline on system. Okay? Let's start with the tetracycline off system. Let's start with this part here. This is our gene of interest. And this the TSG101, our gene of interest, is controlled by the tetracycline response element. And you can see that if the tetracycline transactivator, this guy here, binds to the response element, we will have expression of our gene. Yeah. Now, we have to make our TTA, the transactivator. The transactivator is coded for by this gene here, which will produce the TTA. And the production of this TTA, the expression of the TTA, is localized to the mammary gland. This is where we use our MMTV promoter to localize this to the mammary gland. So we have expression of this all over the place, right, in the entire mouse, but only in the mammary gland. We get our TTA, the transactivator, so we only get expression of TSG101 in the mammary gland. Now comes the cool part. Up here, this little tiny molecule in yellow that you see here, tetracycline. Tetracycline is an antibiotic. There is a derivative of it. It's called doxycycline, which actually we use more in research than tetracycline. So what happens is you can feed doxycycline or tetracycline to your mouse, and it will bind to your transactivator. And once it binds to the transactivator will change its shape. It will undergo a conformational change, and it will not be able to bind to the response element anymore, and now you can stop the expression of TSG101 through tetracycline. And again, you feed doxycycline to your mice. You add doxycycline to your cell culture, and it should stop, repress the expression of your transgene. Now, this entire system, you can also use the tetracycline on system, where kind of like the same idea, but if you start out here again, our transactivator is now a reverse transactivator. It's an RTTA. And when it is produced by its gene, it actually cannot bind to the response element. Once you add tetracycline, the tetracycline will bind to the response element. Again, we will have a conformational change of shape, and now our RTTA can bind to the response element, and TSG101 will be expressed. So two different systems where you can very easily manipulate, do we have expression of our transgene, yes or no? And actually pretty cool, I think. So tetracycline off, we add tetracycline, turn the expression off. tetracycline on, we add tetracycline, and turn the expression of the transgene on. And that brings me to the summer research that I'll be doing. Again, this will be done in collaboration with the Wagner Laboratory. They will provide the cell lines from this mouse model, because now we have this great mouse model. We know how it's supposed to work, but we got to test this. Okay, whenever you have a mouse model, you have to take some cells out and zoom in and look what is actually really happening. So we're going to start out with cell culture. You all have your own cell lines that you will be culturing. You will know everything about cell culture once we're done. We got to test, does the TED on, TED off system work? So we're going to add doxycycline. We're going to test, do we have expression of our transgene, yes or no? Once we have figured that out, we obviously want to look at receptor expression levels. The receptors that we have talked about, the EGF receptor, the IGF-1 receptor, the ERB2 receptor. This will be Western blot analysis mainly, looking, do the expression levels of TSG101 still correlate with our receptor expression levels. And once we are done with that, we obviously want to see, does this influence proliferation of cells? Because that's really the main thing, right? If we turn off TSG101 expression, can we reduce the proliferation of cells? So that is the main idea, the system we'll be using. And if you have any questions, you can ask me right now. I will be around once everyone is done with their talks. You can talk to me privately if there's anything you need to know. Stop by my office. I'm in Swain Hall, the building under the rainbow, room 114. It's by the nursing. I'm the one sitting with the nurses, the nursing department. Yeah, just stop by, email me if you have any questions. And with that,
Next, in alphabetical order, is Dr. Budenr.
All right, so this is going to be a dramatic shift. So I'm Dr. Budenr, Drew Budenr. I work in the chemistry department, so I'm sort of biochem adjacent, let's call it. So the work that I'm doing is the development of these Prussian blue modified biosensors. So I have two projects. There's also a beer project, which everybody's excited about. You hear about it. So what is a biosensor? For our basic parameters here, and just a very flyover explanation of what a biosensor is, it's a sensor, electrochemical sensor, that responds to a biologically important chemical, right? So we all are familiar with the blood glucose sensors. Everybody tests your blood. That is a biosensor. So it's an electrochemical sensor. We're measuring the amount of voltage given off as the glucose reacts in that system. So we have two different basic varieties of these electrochemical systems. We have the direct reaction with the molecule, where that molecule that we're interested in, that biologically important molecule, reacts or oxidizes or reduces to give us a signal. Right? As you can imagine, that number that's capable of being electrochemically signaled, at least produce a signal, is fairly small. So we're typically doing what we call a secondary biosensor, where we use some sort of a I can't talk today. I'm getting all excited. The molecule that we're interested in reacts with something, and then we look at the product of that reaction and look at that electrochemistry. So in the case that we're interested in here, it's typically an enzyme. So the molecule is going to react with an enzyme. The enzyme is going to spit something out. Right? I'm a chemist, so that's what they do. They spit stuff out that we can then oxidize or reduce. And like I said, the glucose sensors that we're all familiar with in the sort of the realm that we're interested in this research is that way. Glucose interacts with glucose oxidase to produce hydrogen peroxide, which is electrochemically active. We can get a good electrochemical signal from the amount of hydrogen peroxide. So as the amount of glucose that hits that sensor goes up, the amount of hydrogen peroxide that produces go up, the amount of current that we generate goes up. So we're sort of modeling it on this system, and we're really trying to interested in finding a new way to make these better, cheaper, faster, stronger, all of that fun stuff, right? and this is probably poorly placed because I didn't know where to put it, but we're interested in something called the way to stabilize our electrode and hold our enzyme through what we call zero gels. A zero gel is just a gel, so it's a inert sort of, it starts out as a sol gel, something that's sort of gelatin, but it's got a whole bunch of solvent mixed in with it. Highly porous, very regular structure. A zero gel is just what happens if you let that bad boy dry, right? Don't really control how it dries, although that's not completely true, right? But you let it dry almost completely. What's remaining from that sol gel is called a zero gel, right? And it maintains a very high porosity, so 15 to 50% of that gel is actually free space, empty space. It has a corresponding high surface area, 150 to 900 square meters per gram of material, and those pores are very small, right, and fairly regular depending on the gel you make. So we have a lot of the ability then to control what gets through this gel, which is what makes it interesting for our work. Right? We're able to form it fairly readily, and it has very sort of happy properties that we like. It's porous, so we can get stuff to go through it, which will be important later. But when we talk, the title is about Prussian blue biosensors. So this work about Prussian blue was first developed or first looked at by a scientist, Karoline, and his group in 1995, specifically because he was interested in replacing these glucose sensors. Right? So it's been since 1995 we've been working with this Prussian blue as an electrochemical sensor, hopefully to get towards this biosensor. And this is the chemist in me had to put this up. Right? It's a 3-electrode system. There's a working electrode, a counter electrode, a reference electrode. We're just going to hand wave through that. Right? The important part is for hydrogen peroxide, we can get detection limits with this Prussian blue sensor of about 10 nanomolar, right, which is extremely small. So if you don't know what Prussian blue is, it's one of the first characterized chemicals. Like when we first started characterizing chemicals in science, the first one they chose was Prussian blue because it gives us this really nice, deep blue color. And it is this regular repeating matrix of 2 irons that are combined through the cyanide group. So we have 1 iron here that has a +2 oxidation charge, and another iron in close proximity that has a +3. +2 and +3 oxidation. So it's capable of transferring electrons fairly readily. You can do up to 4 electrons at a time, which makes it awesome, and it actually changes color. I don't know if I kept that slide. Yep. So if we look at this Prussian blues here in the middle where we have 1 iron that's a +2, 1 iron that's a +3. If we make them both +3, we call that Prussian white, and the color sort of goes away. If we make them both +3s, +2s, excuse me, then it turns green. It's this nice green Prussian green color. We're operating here. But it's a repeatable system, so it can go back and forth without any loss of sort of character. Makes sense? Which is important for us in the electrochemical standpoint. Blah, blah, blah. We mix some stuff together, we throw electrons through it, Prussian blue sticks to our electrodes, we stick it in the oven for an hour and it dries, gives us a fairly stable electrode. This is sort of one of the setups we can use. I'm not going to bore you with the details yet. But what I can say is that we can deposit this in 2 different methods. Because of the way it's created, we grow these crystals, we have control over how much of the surface of that electrode we can coat. So we can either do what I call a single layer, which means that the surface is completely covered with Prussian blue, right, by just sort of zapping it with electrons for some amount of time, right, depending on the size of the electrode surface, that needs to be longer. For the surfaces we use, which are fairly small, about 30 seconds gives us a complete coverage of this electrode. Right? So if we have our electrode surface here, glassy carbon, boom, completely covered with Prussian blue. Right? I call it a single layer because it's a single layer all the way through. You can control how thick it gets by how long you oxidize it or let it run. The toys, in order to characterize that, are expensive, so we don't do it. We just like put a whole bunch on there and be happy. Right? The other thing we can do is what we call a nanoarray. So instead of completely covering our surface, we put small pockets of this Prussian blue around the surface of our electrode. Right? So we get these small pockets. They're sort of, when we do it this way, it's randomly arranged. The size of those little pockets or islands are various. They're not cool circles like I drew here, but regardless. And we do that just by changing how fast we grow that solution. Right? And this has an effect, as you would expect. The amount of Prussian blue is going to give us the information about how readily it reacts. So we can get detection limits. This is a student of mine from the past. His calibration curve got detection limits around 10 to the minus 8th molar hydrogen peroxide, which is pretty good. With the nanoarray, we can go down. You can see it starts to fall off here at the 10 to the minus 8th, minus 9 molar. So that nanomolar to the whatever tens of nanomolars range, we can get very reproducible electrodes. They're very sensitive. Right? Great. Sounds awesome, right? But there's a problem. Right? There's always a problem. Right? Gives you jobs. Right? The problem is that these sensors, the way they act, they don't last very long. Right? Which isn't a huge problem if you're talking about a one chance glucose sensor, right? But if you're doing something else, like monitoring the levels of hydrogen peroxide in a mouse brain, for example, right, you want them to last longer than 10 minutes. Or at least the mouse probably does, right? Because then you have to put another one in. So there's this problem with stability, which has sort of been the bane of my existence for the last little bit of time. Right? We run it through the process of hydrogen peroxide actually reacting, makes it soluble. It doesn't really break down. It just dissolves and goes away. So the way we always overcome this is by covering it up. Right? Let's just seal it in there. And we've seen a lot of ways to do this. You know, the Nafion, which is an ion exchange membrane, Sol gels, which is a zero gel is. There's been work, you can cover it with some other organic polymer to make Wakefield happy. Right? There's some other, you can change one of those metal centers to make it something else. And that is shown if we do a layered approach where we put Prussian blue, this other material, Prussian blue material, several layers of that, it makes it far more stable, but we do lose some sensitivity. Oh, Prussian blue covered it with something else. So, we're doing a twofold attack this summer. This is like sort of the shotgun approach to electrochemistry. We know that these zero gels work well, right? They're fairly easy to produce. Right? And the literature says you can do it simply, right? So what we would do is we would cover our server. That's going to be one thing we do at the end. But we're going to spend some time working with this Prussian blue matrix to see if we can get it to be just as sensitive, but not lose sort of that lifetime, so extend the lifetime of it. And one of the things we're going to do is we're going to take those two methods that we saw worked, covering it with this other hexocyanoferrate, and this idea of a nanoarray, we're going to combine them. We're going to deposit Prussian blue through some sort of screen, some sort of regular matrix, so we get a regular repeating nanoarray where we have nanomaterials or nanoparticle size Prussian blue across our surface. And then we're going to go back and replace the empty space with this nickel hexocyanoferrate. Really to see, so we're going to have, so we put in our Prussian blue, go back, fill in that empty space with this nickel hexocyanoferrate, which I made red here. No, I actually remembered I put it on there. Right? But it's actually green, but green and blue sort of blend together, so we get red here. Right? So hopefully the presence of this other nickel hexocyanoferrate, so in the nickel hexocyanoferrate, which is just fun to say, right, we've taken one of those irons and we replaced it with nickel. Right, which means it maintains sort of that same three-dimensional structure, very defined crystal lattice, but has the effect that it's no longer reactive to hydrogen peroxide. Right? So we can, hopefully this presence of this nickel hexacyanoferrate is going to stabilize these Prussian blue nanoparticles that allows us to have a very sensitive and stable surface upon which we're going to throw this zero gel containing glucose oxidase. Right? So that you can picture, see if I can draw this in 3 dimensions. Our Prussian blue, our electrode here, should have made it a smiley face. Right. Prussian blue, nickel hexacyanoferrate, upon which we're going to cap this whole thing with this zero gel that has embedded in it glucose oxidase. So that we can put a glucose-containing solution on top of this electrode. The glucose would then react with the glucose oxidase, produce hydrogen peroxide within this zero gel, which then will diffuse down and, happy day, react with our electrode surface. Right? So that should be the signal we get out from our electrode is going to be proportional to the amount of glucose we have in here. Right? This has been done, it's just not with the Prussian blue. Like you can stick gold nanoparticles in there, it works hunky-dory, delicious. So we're just replacing the gold nanoparticles with this Prussian blue. So that's one aspect we're going to try. As I said, the other way that they've done this in the literature is they take and embed within this zero gel, you embed not only the enzyme, the glucose oxidase, but you put some sort of a other metal nanoparticle. So in the second design, what we're going to do is put our Prussian blue down. Actually, that's not true. We're going to completely cover our zero gel that contains our glucose oxidase. And in those regular repeating pores, we're going to then deposit these nanoparticles of Prussian blue. So that inside the zero gel, trapped, if you will, we're going to have both the glucose reacting to form hydrogen peroxide, and then the hydrogen peroxide reacting to form electrons. And then since this is a regular sort of repeating conductive matrix, whatever electrons are there are going to then flow through, and we're going to get a very similar system where the response of our electrode is going to be related to the concentration of glucose. Does that make sense? The beauty of this is if this works, then why do we stop with glucose, right? We all know there's like a gajillion, as a chemist, enzymes out there that give us hydrogen peroxide as a result. Right? So we can, if this works, then pick and choose with some degree, our enzymes that we use, and we can have an electrochemical sensor for a wide range of different bioimportant molecules. Mm-hmm. Questions about that? Threw my glucose in there, sorry. All right, so if you have questions about that, I talk really fast, especially when I get excited, and electrochemical cells excite me. Feel free to send me an email or stop by. I'm also in Kesh, but my research lab's over here, so feel sorry for me. I'm in 215. You'll recognize me. If you come up the stairs on that side of the building, you'll see me because I have an office of all windows, and there's a turtle in it, so I'm the person sitting behind the turtle. But if you have questions.
Okay. It's great to be here. I'm Scott Parker. You might know me as the snake guy, as the lizard guy, as the guy who jumps on snakes or lizards in the field, random critters, and gets bit. That's all true. I am that person, but I also study developmental physiology. That's most of what my research is studying the embryonic developmental responses to changes in the environment. Also, during my postdoc, I worked in a reproductive physiology lab at University of Sydney in the medical school. So I have some experience with the biomedical discipline, although most of my research is more directed towards environmental physiology. So having said that, fasten your seatbelts because here we go. All right, so we're going to talk a little bit about reproduction. It's somewhat like Dr. Budenr. I get really excited when I talk about or think about reproduction because I'm a reproductive physiologist. So we're talking about pregnancy. So pregnancy in mammals consists of 3 processes: fertilization, implantation, development, and this consists, and this is important, I can't go into too much detail about it, but there are 2 processes with development that consists of somatic growth, or growth in body mass of the embryo, as well as differentiation. So that's anatomical differentiation, anatomical growth of various structures, like production of limb buds, the jaw, eyes, things like that, organs, organogenesis. So both of these parts or components are important for development, and then parturition, so that's birth. So of course we start up here with fertilization, the union of the DNA of the eggs and sperm, a blastocyst in mammals, then implants into the uterine wall, so there's a little blastocyst. It burrows down into the uterine epithelium, and then in order for the pregnancy to be successful, it has to make connections, establish a vascular network. It has to make connections with the uterine blood supply in order for it to obtain enough nutrients and oxygen for it to grow throughout that developmental period. So this diagram here then is showing the invasive nature of the placental development that occurs in mammals. That's not the case with all vertebrates, but mammals have this very invasive nature of placentation. So here, this embryo is trying to access the oxygen supply of the mother. Okay, so if everything goes successful, then ta-da, your life changes forever, right there. So something that you might not appreciate is that the environment inside is much, much different from the environment outside. And sometimes this is referred to as the uterine environment is referred to as the Mount Everest in utero. And to explain why this is, if we think about what partial pressure of oxygen that we're existing under right now, and in physiology we talk oxygen concentration, express it in terms of partial pressure, which really refers to the packing of gas molecules in a given amount of space. So at sea level, we're basically at sea level, 40 feet above sea level here. We're breathing 160 millimeters of mercury, partial pressure of oxygen. All right, so that's pretty easy to live with. Now, if you go to Mount Everest, the partial pressure of oxygen at Mount Everest is 54 millimeters of mercury, partial pressure of oxygen. By the time that partial pressure is reduced inside our sac-like mammalian lungs, we don't even have enough to serve our metabolic demands at rest. Now, let's think about what happens in utero. So here's a fetus. And in utero, the partial pressure of oxygen that this fetus is experiencing is about 25 to 40 millimeters of mercury. All right, so that's even lower than the ambient oxygen partial pressure at Mount Everest. All right. Your normal arterial blood partial pressure of oxygen as you are sitting here at rest is about 100 millimeters of mercury. So fetuses, embryos, are living in this environment which is very depleted in oxygen relative to the environment. So there's not a whole lot of wiggle room here if there's any interruption or any compromise in the availability of oxygen to the embryo in utero. Right? They're living kind of on a razor's edge. And of course, this is one of the triggers to birth is when the oxygen demand of the embryo or the fetus exceeds that which the mother can supply, then it's time to go to the outside. So I'm going to be looking at this summer these kind of 2 types of hypoxia. So hypoxia meaning oxygen limitation. One is hypobaric hypoxia, and that really refers to global hypoxia. So if we were to hook up a giant ventilator pump to this room and suck all the air out, we would be experiencing hypobaric hypoxia. The other alternative is regional hypoxia. So that would be a limitation on the respiratory membrane surface. Right? So as far as the organism that's dealing with this, it's oxygen limitation, right? The fetus is experiencing oxygen limitation in both of these pathways, but the response might be different, all right? And as far as responding, although we don't want to have a fluctuating oxygen environment in utero or in an egg for that matter, it happens, right? And so embryos and fetuses have to be able to respond to fluctuations in oxygen levels in order to be able to survive during pregnancy. All right, so what are some examples? Well, hyperbaric hypoxia can occur obviously due to high elevation. And hyperbaric hypoxia is one of the main reasons that humans that have evolutionary origins in low elevation environments cannot survive and establish populations at very, very high elevations. Right? So this is one thing that stopped the Spanish from conquering many of the Incan civilizations up in the Andes, is because they couldn't survive and reproduce up there, okay, because of oxygen limitation. We can live and breathe, but as far as reproduction, the fetus can't survive that very low partial pressure of oxygen at high elevation. More with regard to health, the effect of smoking and carbon monoxide, in essence, subjects the fetus to essentially hyperbaric hypoxia. On the other side of that, the other pathway, regional hypoxia, this could be due to preeclampsia. This is a condition where there is malformation of the uterine spiral blood vessels, and that causes an increase in pressure, so high blood pressure. And due to that, there can be regional reduction in oxygen partial pressure or oxygen delivery to the embryo. Placental abruption, again, a reduction in the surface area. Again, the placenta is the respiratory membrane of the fetus. That's how it's getting its oxygen. Its lungs aren't functional, so it has to get it from that placenta that's implanted on the wall of the uterus. Or restriction in umbilical blood flow. So some infants are born or develop with a 2 versus a 3 vessel cord. I have one of those. My son only had a 2 vessel cord, and so they don't have the blood supply that is really required to support their normal rate of growth during development. So all these are kind of examples of regional hypoxia. Consequences are fetal growth restriction, impaired brain development, and of course, if it's bad enough, death. So again, the question is, though, in general, is how do embryos respond to these different types of hypoxia? And this picture illustrates, right, the difference in size. Right? So this is a infant that was born with fetal growth restriction, and here's a normal size one. That looks like a pretty big baby, but nonetheless. All right, so. But we're not going to be working with human embryos for obvious reasons, and neither are we going to be working with mammalian embryos because of to work with them it would be very invasive, right? Because all mammals except for a couple are viviparous, so it's difficult to study them. Also, there's more ethical concerns, right? So even working with this breaks my heart, lower vertebrates like reptiles, we have to have animal ethics permissions to work with these. So there's a lot of complications in working with mammalian systems. So that's where the egg comes in. The egg is a really wonderful, powerful model. So fortunately, among amniote vertebrates, so amniote vertebrates consist of mammals, birds, and reptiles. We all have these extra embryonic membranes. These are membranes that are genetically part of the embryo. They're tissue of the embryo. And they carry out various physiological functions. Right? So we all have these membranes early on in our life. All right? So you can see that over here. Here's a mammal. Here's a reptile or a bird. All these extra embryonic membranes are present in both taxa. They carry out, in many ways, similar functions. But there could be some structural differences. Additionally, the ontogeny of embryonic development, particularly early on, is very similar among amniote vertebrates. Right? So we all start out with having pharyngeal slits in our neck. We have a postanal tail. we have this notochord, we have all these very conserved, evolutionarily conserved features. And so we can capitalize this and use eggs as a really nice model system for studying some of these fundamental effects of hypoxia on embryonic developmental processes. So the aims and experimental approach. So I aim to test the hypothesis that the hypoxic stimulus, whether it's hypobaric versus regional hypoxia, during embryonic development differs among our selected amniote taxa. And the embryo models that I will use is chicken. So this is a little chick sitting on an ostrich egg. So a chicken egg is really this size down here. Japanese quail, leopard gecko, and crested gecko. Now, I don't have time to go into this, but each of these taxa have different eggshell characteristics in terms of thickness, and that, because of the thickness, that affects the rate of oxygen diffusion. So there's some other interesting manipulations that we can do just by having these different models to work with. So what are we going to do? So one group will treat with regional hypoxia, and so you can do that just by painting a portion, in this case about 2/3 of the eggshell with paraffin wax, and leaving a small portion open that the embryo is going to respire through. Hypobaric hypoxia, we take the eggs and incubate them at experimental oxygen partial pressures, 4%, 9%, 15, and 21, and keep them at constant temperatures that are correct incubation temperatures for the species. So quail and chicken warmer, lizards 28, and we'll impose these treatments kind of midpoint in development, let them go, and then see what happens after a period of time. So after 5 days or 15 days for geckos, we'll measure the embryo stage. So dissect out the embryo. So this is requiring embryo micro dissection. Dissect out the embryo, weigh them, look at the features to determine the embryo stage. Prior to doing that, obviously, when the embryo is still alive, we'll measure oxygen consumption using an oxygen analyzer, heart rate using a little a little heart rate monitor, and finally measure chorionic vascular density with common histology, some typical histology and light microscopy. So these are a couple of my current very fantastic research students. Sierra is with us today. Unfortunately, Robin couldn't be here, but she's with us in spirit. So they're operating a microtome, and what we would do is look at the sections of these of eggs incubated under those different conditions and basically just count blood vessels. So my goals are for students to become proficient in the use of these various techniques, so histology, embryo microdissection, oxygen consumption / respirometry, be able to analyze and interpret the results of these data and present them in graphical and tabular format, and of course be able to present and explain these results in scientific research conference format. So these are just a couple of examples of my former students presenting at venues. And Serena is now a high school teacher down in Georgetown. Kira is got accepted to 4 medical schools, count them, 4 for all you future doctors, is now at the University of Gainesville in Florida. This is my lovely wife's student, so I don't know what she's up to now. Okay, so thanks to everybody for funding and all the support, especially all the tireless dedication of the help that we have in the lab. Couldn't function without all the wonderful student support. And are you ready to start doing stuff? You can do homework and be surrounded with snakes. Okay, thank you. Talk to us.
All right, so my name is Paul Richardson, and basically I work with bacteriophages. Those people who get paranoid when you say you're working with viruses, it's okay. These are viruses, they only infect bacteria. And the motto of our lab is harness evil to fight evil. So we are looking for these naturally occurring viruses to use them to basically treat some problems we have and man has created. So one of the projects we are really interested in is MRSA-based research. Basically looking at bacteria that's resistant to antibiotics and coming up with a different means of trying to cure these diseases. All right? And I'm not going to go through all this, but I just threw up a whole bunch of little places you can go and visit to talk about the relevance of why we need to invest in something like bacteriophage, because of the emergence of all of these bacteria that are resistant to a lot of different classes of antibiotics we have. All right? I mean, over here, 42 per 1,000 hospital cases have something to do with methicillin-resistant Staphylococcus aureus. The number of MRSA cases have doubled from 2003 to 2008. So, a comprehensive study found in 2008 that only 15 antibiotics out of 167 under development had a new mechanism, meaning it was targeting a new pathway to actually attack the bacteria. The rest were using old, just modifications of existing antibiotics to treat these infections. So traditional medicines are not keeping pace anymore. And we have to kind of open up our minds and look at different alternatives. Alternative facts, if you would. Okay, somebody got it. All right, so I'm interested in looking at bacteriophages because, well, quite frankly, I'm lazy. I don't want to invent something new. I just want to kind of go look around what someone's already made and just figure out how to use that. And nature's done a great job of coming up with these things. All right, so what are some of the advantages of using bacteriophages for therapy? Well, one cool thing is they're self-replicating, so we don't have to really worry about dosage too much. We can give it to someone, and as long as there's infectious bacteria, this thing can keep on going. It's a narrow host range, so we can target very specific strains of bacteria and not worry about targeting those bacteria that we need to keep us healthy, to keep us safe. It requires the bacteria to grow and proliferate, so hey, neat little way to do it. It can evolve faster than the bacteria, so as the bacteria tries to mutate and stay ahead, the virus can, of course, mutate and try to see if it can keep up. And once developed, please notice the once, they're fairly cheap to produce. Now, getting to that point is entirely a different story. But now we got to talk about some of the disadvantages. One of the reasons why we really don't have a lot of bacteriophage therapies today was antibiotics are more broad spectrum. Okay? Back around World War II, particularly the Russians had a lot of bacteriophage kind of therapy, but they're very host specific. So that can be a problem because it can be down to different strains of bacteria. So some staph might be destroyed, others proliferate and have no problem. It does require bacteria to grow and proliferate, so we need to use those if we want to make more of these viruses. It must be placed in contact with the virus, and this might not be something you understand, but let's say you have an internal infection of bacteria. You have to somehow get that virus to the wound. And of course, if you're trying to go through all of these testing procedures to basically allow to use a drug, you're going to have to go through all those different scenarios to make sure that it's safe. And it might generate a host immune response. So your own immune response, if it's internalized, could actually come out and start attacking the bacteriophage. So there are some problems. Now, it's not like I've come up with something new and I'm like, hey, this is great. Why hasn't someone thought of this? This has been thought of a long, long time ago, even before I was born and all the other faculty in here, much to some of your dismay. All right? So this is an old idea that's just gaining prominence now because we're having some difficulties with these antibiotic-resistant strains. And I've put these up here to kind of show you a bunch of times where here's a bunch of different diseases and when certain very crafty scientists came up with ways to treat them using bacteriophages. But if you look at these dates, you'll notice these pretty much all are post-World War II. And it's World War II when, does anyone remember the name of the famous scientist who came up with antibiotics?
Mr. Penicillin.
Yes. What is his name?
Mr.
Anyone? Euler.
What to start with?
F.
Fleming.
Fleming. Fleming. All right. Okay. So, Mr. Fleming was the one who came up with antibiotics, and that really showed a lot. It's a lot easier to use, a lot easier. It could treat a lot more bacteria. So it kind of took over, particularly in the Western society. But now over in the eastern regions of this world, particularly the country of Georgia, it's actually quite prevalent. And there's been a lot of research there, kind of way ahead of the rest of us. But I just throw this up here to show you the history of bacteriophage use, and it wasn't just for medicinal value. I mean, it was used a lot for the discovery of a lot of molecular biology. One of your favorite classes, right? No. By the way, he's in the room. I might want to show a little bit of enthusiasm. One thing. Anyways, we move on. Now, in the last couple of years, we've actually seen a resurgence of bacteriophage therapy. There's actually a company up there that's actually doing a lot of work. They got a big grant with the U.S. government developing a lot of phage therapy. They just started about a year and a half ago going into phase 1 trials, doing a lot of these things. So they're moving forward. The FDA has approved bacteriophage as an additive for meat. There are a lot of trials going down. So with all that said, let's talk about what I'm trying to do here. So our lab is involved in using bacteriophages to tackle the issues plaguing humanity. So I got some really neat, creepy pictures from the internet. Does anyone recognize this?
Is that the medicine guy that would like take people?
It's not Sean Connery. It's not Medicine Man. This is not the guy who found the cure for the plague of the 21st century and lost it. Okay, no one got the movie quote. Okay. Yes, but what is the plague? This was those famous masks that the plague doctors, and I always like to use quotations when I refer to them as doctors, treated the plague. Over here, this is the 4 horsemen of the apocalypse. And this is a bacteriophage. I've never seen that one before, but oh well. And this is some art in Italy. It's supposed to represent misery. So, okay. So let's talk a little bit about what I like to do, all right? So what I like to do is I go out and I start collecting specimens anywhere I can. Matter of fact, some of you probably have met some of my research students asking you, hey, can I please swab your nose? Anyone? Yeah? A few of you? Yeah. So yeah, a few of you at least know these. They're going around and they're saying, hey, can I swab you? And they're coming in and they're collecting. Here's some of my old research students. They are swabbing an individual in the nose and behind the ear and collecting specimens, and then we bring them back into the lab. And what we have done is we've developed a whole bunch of PCR-based tests to look for a lot of different types of bacteriophages. Many years ago, we started with just coliphage, looking at water and soil samples. And that was helping us look for, anyone? Indirectly measuring fecal contamination. What was that?
E. coli.
Yeah. And then we've moved into, we got a bunch of Staphylococcus aureus phages, so we can check for them. We've also been moving into bacteria to actually detect, is the bacteria present? So we've developed a whole lot of PCR-based tests. I've thrown up some images showing some of our actual student work. This is some of the first PCR tests we did on coliphages, and believe it or not, we actually were able to detect 1 viral particle in an entire sample. So if that sample has 1 viral particle, we can detect it with these these primer sets we use, and they're targeting glycoprotein 23, which is a capsid protein, and then glycoprotein 43, which is part of the polymerase of T4. Now, I'm going to put in a plug for Dr. Savasco here. You know how we designed these things? Bioinformatics. You go and you look at a whole bunch of different viruses, and then you try to target things that don't mutate. Now, finding a region in a virus that don't mutate is difficult at best. But if you spend a lot of time, you can find them. And then we developed some primer sets to target those sites. And in particular, this GP43 has turned out to be quite impressive. It actually is not strain specific. It is, I mean, it can get many different types of bacteriophages. This one has not proven to be as robust, but yeah, we've kind of given up on it. So let me first talk about our projects. For the first is our MRSA project. And what we like to do is we go out and, like I said, we collect swabs. And then we bring them back to the lab, and then we run two types of tests. The one that I just told you about, PCR, but then we also run Dr. Goulis's favorite test, the plaque assay. Is that not right, sir?
Probably.
Probably? All right, I know it's your favorite. You're just going to come to admit it. All right? And we run those, and we're looking for, hey, do these people have it? And now, I have some bad news for you people. We have done this test we've done these studies for over 4 years, and we've come to one conclusion. Something is happening after Christmas break. And all of a sudden, we go from no phages on people to over 25% of the population of students have colophages, which is phages against E. coli on their face. And 33% of you have colophages phages against staph. So we are going to try to figure out what exactly is going around spring break. I know we just got a grant, so we're going to start looking at measuring individuals over the semester to see what is going on with the population dynamics on your faces. Because, wow. And what really scares me is the colophages.
Yeah.
It scares me a little bit what to say how that is happening. But I would let you know if you ever see me in the restroom, boy, do I wash my hands. I really wash my hands a lot. Bigly. All right? Now, the next project we have worked on, which has had, so great success, is the unnatural amino acid project. Now amino acids can be in two configurations. Dr. Wakefield, what is the one that's the most biologically active?
Should be L.
That is correct, sir. Awesome. So what we do is use D because they're not really natural. And we're trying to see if we can use these unnatural amino acids to inhibit bacterial or viral growth. And I have to admit, we haven't really gone anywhere with this. And it really has to do with getting the techniques right. A group out of Harvard came up with this right before we did. And they have all these nice sophisticated pieces of equipment that cost $500,000 to monitor these things. And we're using a $1,500 UV vis. So, But anyway, so one of the projects we're trying to look at is using some of these D amino acids and can it inhibit bacterial growth? Because like I said before, there isn't a disease I like, right? I mean, I constantly talk about killing things. And most of it is microorganisms, viruses, bacteria, whatever. But, that's what we're trying to do. Now, the last project, which is by far the most important, is our fingerprinting project. It's one thing to collect all these things that are growing on your face, but I'm having a problem understanding if the thing growing on your face is the same thing as growing on your face. Though my PCR primers might say, hey, they're there, I'm not really certain what's there. So we're trying to come up with a way where we can get the viruses and isolate them, which we've done. And then we grow them up to big quantities, which we've done. And then the part we're having problems is how do we separate out the DNA of the viruses from the bacteria we grow them up in, and then trying to do the restrict and digest to create a fingerprint. Okay? Now, some of you might not understand what that means, but if I mention something like Maury Povich or Jerry Springer and who's the daddy, then maybe you might kind of understand. But we're trying to create a genetic fingerprint so we can compare and contrast these viruses we're collecting. So I can know whatever was growing on your face, that happens to be the same growing on his face and her face, but not that face. And we're trying to also figure out, are these things coming from the environment, or are they coming from just each other? So we're trying to, because we're also testing soil and water samples, but that's not really what we're going to be doing this summer because that's not biomedical research. That's more environmental, and that's the other branch that my lab does. So I want to thank you for your time, and if you have any questions, when we're all done, please come talk to me or come talk to the other 3 fabulous research scientists that have come out to share their experiences with you. Thank you.